as usually we do for this webinar, the webinar are registered. So I just now started to register the webinar. So I inform everybody that the webinar is registered. So you have to agree about this. The author already agreed to have recorded their presentation. The recorded presentation, the recorded video will be published on the iPlanta webpage. Well, we are at webinar number 9. It has been a very long work we have done until now in the last 3 months, and we were just now commenting with some colleagues that finally it has been really a nice opportunity to work on the webinars, because we were forced, because of the COVID pandemic, to move from the in-presence meeting to the online meetings. And so we decided to use this technology for enlarging the network, and that has been a positive thing, because now we've reached practically 900 contacts for this network. So we enlarged really a lot the network related to the iPlanta, the number of people that have been following all this activity. So in some way, finally, we were able to bring near to the hand this cost action by enlarging the network, by discussing several topics, and trying to understand even much more and disseminate even much more all the potential benefit of this technology. And today webinar, it's another important specific aspect of this technology. So what is the impact of what can be the role of RNA technology in also at epigenetic level, or things that we need to keep in consideration if we want to apply this, if we consider RNA technology in our study. So, well, this is just my very short preliminary introduction, and I want to thank today's speaker, and in particular Athanasios Dalakoros, that it's thanks to him and Michael and others that this meeting happened. And so I ask Athanasios to take the floor, to take the manage the program of today, introducing the speakers and managing the presentation. I anticipate again that during the webinar, people, if you have question, you can post your question on the chat. Athanasios can take the question to the speakers, but then at the end we will also have space for a discussion. Okay, thank you. Athanasios, it's your job now.
Okay, thanks, Bruno. So good morning, everybody, from me as well. And today I think we'll have an interesting webinar, which will last approximately 3 hours, if I'm not wrong. We'll have 5 talks, and which will presumably touch upon the tripartite interaction between epigenetics, RNAi, and development and plant development. We know that, I say interaction because we know that RNAi, especially small RNAs and microRNAs, regulate plant development. We also know that epigenetic events like DNA methylation or histone modifications also regulate plant development. And vice versa, we know that RNAi and epigenetic events are tightly connected, I mean, mechanistically. So it's not easy to separate epigenetic events from RNAi or vice versa. So I don't want to spend more time on this. We will hear the talks. And first we'll start with Michael Vassenager from iPlanta Institute in Neustadt, Germany. And I would like to remind the audience that Michael was the first to discover the RNA Direct Dna Methylation in plants back in 1994. So it will be a great pleasure and honor to hear what he has to say. Michael.
Well, thank you, Athanasios. First of all, we are no longer the iPlanta Institute. We are only agroscience. So both departments fused now for the future. And thank you for your nice introduction. And I'm very happy to see you as a chair since we had a long period working together. And I think it's a great pleasure to see you here as a chairman now. Thank you. Let's get started. I will share my screen. Everything okay? You see it? Do you see my screen?
Yes.
Okay, great, thank you. So as Thanasis already indicated, this is the paper since 1994. I'm busy with analyzing this strange mechanism of RNA Direct Dna Methylation. It was actually my first paper, and it started, of course, my career for the last 30 years. I'm still busy on it. Okay. Let me start with what epigenetics is involved in. Other processes too, but epigenetic is one of the major mechanisms that helps to get a gene regulated. If we have a cell, we have the genes in there, and the genes can be in an open structure or in a tight structure, depending on the histone modifications. And we have the active genes and the inactive genes indicated here in green and red. And in this, the cell is one cell of a leaf, for example, and you see that all cells contain identical information that is encoded by genes, but in different cell types, different genes are expressed, and the combination of active genes determines the cell type, which we call differential gene expression and gene regulation. And as I said, one major issue or one mechanism that is involved in that is I will go down. I have a is the DNA methylation. Although histone methylation or modifications are absolutely on the same level as DNA methylation, in plants we have the advantage that in general, methylation precedes histone modifications, which is different in mammals. That's vice versa there. But since we have here first the methylation and then histone modifications, we can easily analyze the methylation patterns of a genome today. What is very important for understanding the gene regulation is the difference between the so-called symmetric methylation, which can be maintained after cell division because it's due to this symmetric CG. There is an enzyme in mammals and in plants that can recognize the CG and methylates the opposite side. And this is how it can easily be given from one cell to the next cell, and maybe also from one generation to the next through the progeny. This is different with a quasi-symmetric, which is only present in plants where you have a C and then an H, which is not a C or a G, which is maintained very inefficiently. It's a very particular thing. But the most important is the asymmetric methylation. If you see after cell replication, you have no C or no indication for any enzyme what to do on the opposite strand, because here is no C and here is a G, and there is no information for this C. And this can be only maintained or introduced by the de novo methyltransferases, and it requires always the trigger to be present for methylation. We did this here as an example. It's not important how we introduce this methylation patterns here. It's by Rddm, and you see that it's a very, very efficient high methylation. But if we segregated away the trigger, then in first generation this is maintained, and the second this is maintained. And as you can see, it's only CG methylation that is maintained here. But you may now consider that if you have the trigger in, you may regulate genes by this high or this dense method, de novo methylation, but it will disappear in the after cell division if the trigger is no longer present. And this allows you a nice regulation, epigenetic regulation within one cell. And this probably, this is sufficient for a regulatory element to get downregulated because of this methylation, then you can maintain it once, even if the trigger is out, it will stay and stay and stay and stay. And this we can do for millions of years. There is a nice paper from last year where they could show that if you lose the de novo methylase function, the maintenance function can keep on with the dense methylation here for millions of years. And I think this is something where you see what is inheritance, how stable it can be, only side effect. And now we can ask, what is the connection, as Thanasis already mentioned, between RNAi and the epigenetic modifications in plants? This, I don't need to introduce all these pathways for RNAi, you know. This is the well-known pathway for RNAi mechanisms, and this here is the pathway for the chromatin modification, the DNA methylation and chromatin modifications. This was the findings from Peter Meyer in, I think it was 1993, when we had this nice petunia that were deregulated by a epigenetic mechanism. I don't want to bore you with this most recent cartoons of how Rddm works. I do not agree with all these steps, so I would like to only give you a simplified model, which is RNA. It must be double-stranded, long or small. It can come from polymerase, from polymerase 1, 2, 2, 3, 2, 4, and 5. It may come from RDRs, RNA directed RNA polymerases, or in vitro transcription of inverted repeats, virus replications or viral replication, doesn't matter. You need double-stranded RNA. And then you have the de novo methylase, the mainstream range methylates, that needs probably some additional proteins to act on the DNA, which needs strand separation and nucleon dissociation. And then you have the de novo methylation, whether this is on both strands simultaneously or not is not known, and whether the maintenance function of the DNA methylation is also already activated at this stage is also not known. In addition, we have something like that we also know from RNAi, the self-reinforcing loop, because if the DNA is methylated, polymerase 5 is transcribing this methylated region, and together with AGO and some 24, some small siRNAs, not the 21s, not the 22s, but the 24s, bind to these AGO proteins, and then with the help of polymerase 2 and probably RDR2, you have a secondary double-stranded RNA that feeds into this pathway, and then you have this self-reinforcing loop. Okay, tools for the induction of epigenetic modifications are, in the first view, chemicals like 5-azacitidine or seborrheic. And if you apply this to the plants here in Arabidopsis, you see really severe growth defects in the seedlings. And if you look for transcription of some genes, you see that some are upregulated, also some are downregulated, and transposons become activated. And this is simply by changing the methylation pattern, because these cytidine analogs can inhibit the methylation of this DNA. Okay, both treatments induce unspecific DNA methylation. Very important. You cannot predict what region is first demethylated, and so you never know the outcome of this. We found a way with a transgenic plant that is very specific. If you introduce into an intron of a gene an hairpin RNA, you end up with a double-stranded RNA that is retained in the nucleus because it's in the intron and does not go. If you do it in a gene, then it's pathing to the cytoplasm probably and is not that active as in the intron. And with that, you can get very efficient methylation and specific methylation as indicated here. If you see, this is the GFP intron and a promoter region of a CPM CMPS promoter in an inverted repeat, and the rest of the intron, and the GFP, the 3' end of the GFP. And if you see, this here is the GFP, this is the start of the intron, and this is the hairpin. And precisely at the cloning site here, we found dense methylation, almost 100% methylation in this region. In the spacer here, this part, we have no methylation. And again here, methylation, dense methylation starts, and it stops in the intron output. So very, very specific and very dense. And this also works in trans. When we use this construct for another transgene for methylation, we found the same at the border sequence here. Dense methylation starts. So the black triangles and all black indicates methylation, and the white dots or triangles or squares is non-methylated. With all the controls, just believe me that we have very dense methylation in this part that is corresponding to this trigger here, and all the other is non-methylated. Okay, another tool is the virus-induced Rddm. You can use the virus for PTGS if you put the coding region into the virus vector. This is here the control, no silencing. If you have the coding region in this virus and you infect, then you have non-heritable silencing, the post-transcriptional gene silencing. And if you put the promoter sequence here into the virus, then you have the transcriptional gene silencing, and this is maintained, should be maintained in the next generation. I'm very careful with this because we have not looked into detail whether this is really the truth. Okay, plant viruses do not infect all cells. It's a disadvantage, but virus-induced Rddm does not require the production of transgenic plants. It's an advantage. But now we may, it's also very sequence specific. We can also consider the topical application. You know, our high pressure spraying method works fine to introduce small RNAs into the plants. Also the carbon dot procedure works fine. This was published, and these are our own data. It really works very nicely with carbon dots. And we have, up to now, maybe Thanasis knows more about this, had some more data on this. We do not know whether this can induce also DNA methylation, Rddm, but we are just analyzing it. We have some indication that indeed this might work. So if this is true, we can use the topical application of RNA molecules to trigger Rddm of a specific sequence. Okay, the third, and this is no longer related to our RNAi story, let's say it this way, but you can modify, of course, the epigenetic by plant epigenetics by using alternative methods like CRISPR-Cas. You can knock out genes that are involved in de novo methylation, demethylation. By the way, the Ros gene here is one of the genes that becomes activated if the regulatory element of this gene is densely methylated. The general rule is if the promoter or regulatory elements are methylated, then the gene is shut down. But this is upregulated upon methylation. These are all enzymes that are somehow involved in DNA methylation, of course, by using CRISPR-Cas or alternatives, tail and zinc finger, you can knock out these genes. You may also overexpress these genes, and you may combine overexpression and knockouts. For example, if you knock out the DRM, you have no de novo methylation, and if you overexpress the ROS1, you have an active demethylation, and therefore you should end up with a completely demethylated gene. Whether this is the plants are viable or not, I doubt. The next, but you can do it in RNAi and lower the expression levels. But you can also use the CRISPR system to not cleave or make genome editing, but to target some enzymes that are involved in DNA methylation or histone modification to a specific sequence. So also this method would be a very specific procedure for changing the DNA, the epigenetic status of the cell. What can we do with it? Where is epigenetic involved? It doesn't matter how you change the epigenetic, it will lead at the end to differential gene expression. This you can analyze by a transcriptome analysis, and you may have a decrease in 5-methylcytosine or an increase, downregulation here, upregulation here, and then you may or you will activate the transposons. If you decrease methylation, they become activated, and you may also activate real, let's say, genes that are downregulated by methylation, by epigenetics. And if you have increased methylation at a certain regulatory element, for example, you downregulate the gene, and this is clearly an epigenetic effect. Whether it's random or a direct insertion or epigenetic effect depends on what is the outcome. A random effect is, for example, increased mutation rate. If you have the cytosine methylated, it frequently, or more or less frequently, shows mutations, a transition from C to T, and then you have a real mutation, irreversible, and you may have gain or loss of function new transposon insertions. The real direct effects, which are reversible, is up or down regulation of transcription factors or up and down regulation of genes, simply genes. Not all epigenetic regulation mechanisms are, as I said, associated with altered DNA methylation patterns, but you can easily analyze the DNA methylation, and it's very difficult to get a detailed picture on chromatin modifications, histone modifications. That's why we prefer the DNA methylation analysis, and we can also easily manipulate it. And the last slide, mutagenesis via transposons would be a nice tool to get new traits into plants, because if you have the transposon activation, you may end up with gain of function. If it's close to in a promoter sequence or close to a gene, it can activate the gene, but it can also, of course, jump into the coding region, and therefore you have a inactivation of the gene. This is genome-free mutagenesis, and then in the next generation, you can, of course, screen for new traits, and they will be stable and will be inherited. Okay. With the introduction of my current group here, I thank you, and I'm open for questions.
Okay, thank you, Mike. So, are there some questions on this presentation?
Not in the chat.
Yeah, not in the chat. Please unmute yourself if you want to ask something.
Oh, well, I have a general question. So among the different solutions that in some way you proposed, including the last one that is kind of traditional mutagenesis that I like, I think that still can be really helpful. What is your suggestion in a more applied way? What can really be more easy to apply and helpful to create maybe new interesting variability?
If you include the GM plants, then I think this would be a really perfect way. We can absolutely specifically methylate a sequence, as you could see. But we may be able also to do it by exogenous application. This has not been tested so far. And we will see if this really works, then you can easily manipulate the plant. The point is, if you have a vegetatively propagated material, potato, for example, you can do it in a shoot, and then it will be stable forever. As you could see with the 50, I think it was 50 million years stable methylation pattern. And therefore, this could be a really nice way to change the trait of a plant without any changes of the DNA. And there's no mutation.
No, for us, well, that's the point. Not only potato, but the apple, I don't know.
Yeah.
That could be.
It was just an example because we are going to work a little bit on potato probably, and therefore I took this as an example. There are many plants.
Thank you. There is a question from Antje.
DNA precedes histomethylation. Does that mean that DNA methylation is a prerequisite? In most, Antje, yes, in most cases it is the case. You will, if you have a methylation in a, for example, regulatory element, not in the coding region. In the coding region, probably there is another way how the chromatin is changed because methylated coding regions tend to be very stably transcribed, expressed. But if you have it in a regulatory element, not only promoter, could be also upstream, then it recruits the histone modification enzymes that also can contribute to DNA methylation at the end. So a kind of reinforcement. But the first is that you have by Rddm, you have the dense methylation pattern, recruiting all the enzymes that modify the histones in a way that you have the heterochromatin or condensed inactivated promoter.
Okay. I have one more question. We have the time. So you showed all these nice figures on the silenced 16 seed leaves, where you use the carbon dots. Have you tried the carbon dots with a long double-strand RNAs?
So far we have not, but it was in one of the papers from the Bayer group, and they said that 50 base pairs still are fine, with 90 they failed, also with the carbon dots. And we are on the way to use now by high pressure spraying and carbon dot procedure the 24 nucleotide siRNAs, because if 22 go in, I guess 24 will also go in, and see whether they could act on the 35S promoter.
Okay, so if we have no questions, no more. Yeah, we have one more question. Maria Ladera says, I'm not expert in the topic, but how can you the gene that you want applying exogenous dsRNA?
Yes, as I just we have to verify this or see whether it really works. But the idea is indeed to take 24 nucleotides along siRNA that is associated with de novo methylation, as you will find it in all the papers. And the major trigger, the scientific community is convinced that these molecules are the major triggers of Rddm, and we will be able to get these into the plant. This is no doubt. Already Thanasis did it also with high pressure spraying. The effect, we have not analyzed the effect on methylation at that time, and we were interested on post-transcriptional gene silencing, but now we apply it by the two methods, as I said, and target a promoter instead of a coding region, and the 24s instead of 22s or 21.
I think we'll hear also in my talk some data on this stuff. So I can, we have no more questions. So I can start sharing my screen.
Alexandra was also writing something.
Okay, yes. Alexandra says, do you know examples when an increase in DNA methylation would induce its expression level? So whether a hypermethylated gene with...
Yes, I tried to put it in my talk. Maybe it was not so clear, but the Rnas gene, the demethylating enzyme, this is a nice balance between de novo methylation and demethylation. If you have a heavy de novo methylation, the Rnas promoter will be methylated and becomes activated, and this enzyme demethylates the DNA. So it was nicely, two papers that nicely validated, verified that upon increased methylation, this promoter becomes activated. So far, this is the only example, but I'm pretty sure there will be others. We have to find them simply.
Okay, so we will continue with my talk now. And okay, so in the next half hour, I will discuss about the so far less investigated issue of whether exogenous application of RNA molecules trigger epigenetic modification in plants. So I will give a short introduction of RNAi because perhaps not all people are familiar with it, but so very briefly, double-stranded RNA triggers RNAi in plants. The long double-stranded RNA is cleaved by endonucleases called DCLs. So DCL4 cleaves the double-stranded RNA to 21 nucleotide small RNAs that recognize homologous mRNA and cleaves it. And this is called PTGS, or as Michael said, or post-transcriptional gene silencing. Now, DCL2 may also cleave the long double-stranded RNA into 22 nucleotide small RNAs, but they also recognize homologous mRNA, but seemingly they do not cleave it, but it seems more likely that they recruit RDR6 to the 3' end of this RNA and generate secondary double-stranded RNA and secondary siRNAs. And finally, DCL3, which is localized in the nucleus, is cleaving the double-stranded RNA into 24 nucleotide small RNAs, which are loaded onto AGO4 and migrate back to the nucleus, where they recognize DNA, directly the DNA, or a nascent transcript produced by polymerase 5 in order to methylate this DNA. This is called, as Michael said, Rddm, or RNA Direct Dna Methylation. So there are the widespread assumption is that that the small RNAs are triggering Rddm, and there are two models, or there are two pathways of Rddm. The canonical Rddm, which is supposed to be triggered by 24 nucleotide siRNAs, and the non-canonical Rddm pathway, which is triggered by 21 or 22 nucleotide small RNAs. Very briefly, according to the canonical Rddm pathway, the DNA that should be methylated is first transcribed by polymerase 4, and this transcript is processed by RDR2 into double-stranded RNA, and then this double-stranded RNA is cleaved by dicer3 to 24 nucleotide siRNAs, which are loaded onto AGO4 and recognize a polymerase 5 transcript in order to methylate the DNA. Then in the non-canonical Rddm pathway, here we have transcription by polymerase 2 and not by polymerase 4, and this transcript is cleaved and then copied by RDR6 and not by RDR2 like here, into double-stranded RNA. And presumably here many dicers are involved, which generate 21 and 22 nucleotide small RNAs, which seemingly are loaded into AGO4 and again recognize polymerase 5 transcript for methylation. However, these two models, both of these two models presuppose that dicer produced small RNAs trigger methylation. But it seems that it's not really the case, because there have been several reports challenging this assumption. For example, it has been found that in a quadruple dicer 1, 2, 3, and 4 mutant in Arabidopsis, Rddm was not abolished, indicating that siRNAs are not absolutely required for the establishment of Rddm. Then we also know that AGO4 is also not required for Rddm, and also that polymerase 5, which is here produces the scaffold transcript for methylation, has been shown to be recruited to an already methylated DNA locus. This data suggests that perhaps these pathways that we see here do not reflect the very first steps of de novo methylation, but most likely reflect the amplification of an already methylated locus. So, this assumption is strengthened by some previous data we have, where we found that DNA can be established in the absence of small RNAs, but in the presence of homologous double-stranded RNA. So our assumption that we had some years ago, Michael Vassenager, was that the de novo methylation, the very first steps of de novo methylation are triggered by long double-stranded RNAs and not by small RNAs, regardless of their size. So to be more precise, if here we suppose that this is a non-methylated, completely non-methylated locus, and we have a double-strand RNA present in the nucleus, then the double-strand RNA will recruit the methyltransferase DRM2 to this DNA, but we assume that the methylation that will be induced in this locus, perhaps it's not perfect, it's incomplete. Nevertheless, to this incompletely methylated DNA, polymerase 4 will be recruited and transcribe, produce small transcripts of 40 to 50 base pairs. This has been shown. And this short transcripts will be processed by RDR2 into double-strand RNAs that will be cleaved by dicer 3 into 24 nucleotide small RNAs. And then these 24 nucleotide small RNAs will recruit once again the de novo methyltransferase DRM2 to the DNA for methylation. So here, this model presupposes that the polymerase 4 is recruited to a heavy methylated DNA and also polymerase 5 to a heavy methylated DNA, which fits with the previous data. So first with the novel methylation induced by double-strand RNA and by the amplification of methylation induced by small RNAs, the target locus in the end will be completely methylated at any sequence codex, CG, CHG, and CHH, as we can see here. This is the asymmetric methylation, as Michael said. Now, in the next generation, or after meiosis or mitosis, and in the absence of RNA trigger, the asymmetric methylation will be lost. But the CG methylation, the CHG methylation will be maintained, even if no RNA molecules are present, but by the action of methyltransferase 1 and CMT3. So this is important to remember that while at the absence of RNA molecules, asymmetric methylation is lost, symmetric methylation and quasi-symmetric methylation can be maintained. So why do we care about this? I mean, if we discuss about exogenous application of RNAs in plants, so why this is important? So most applications in plants, when we spray or inject RNAs in plants, the rationale is to trigger mRNA degradation of a given gene, whether it's a plant gene or fungal gene or viral RNA or whatsoever. However, when we apply, for example, double-strand RNA in the plant, then this will this will be processed to 21 nucleotide small RNAs and trigger PTGS in the cytoplasm, but it might also be transported to the nucleus to trigger DNA methylation, as we said before. And this is, in my opinion, quite critical, because if we suppose that the RNA sprayed plants are GMO-free, and they are GMO-free, we have to take into consideration that perhaps they are not epigenetically modified free, because they will have some epigenetic modifications. And if we take into consideration that the gametic cells are produced from somatic cells, then these modifications may pass over to the next generation. So let's see now what has been, what we know so far on application of small RNAs. I will start with the spraying or exogenous application of 21 nucleotide small RNAs. And these small RNAs are designed to target an mRNA, so they map to the coding region of a gene. So what has been shown so far is when you apply 21 nucleotide small RNAs in plants, then they are loaded onto AGO1, and they recognize homologous mRNA for cleavage and PTGS. And this has been nicely shown several times before. What has not been tested, but it seems unlikely, is that this 21 nucleotide small RNA will go to the nucleus to methylate the coding region. I think this will not happen, but it has not been tested. Then when we spray 22 nucleotide small RNAs that again match the coding region, the 22 nucleotide small RNA will be loaded onto AGO1 and recruit, as we said before, RDR6 to this target, especially if this RNA is transcribed from intronase gene. And then it will generate secondary double-strand RNA and secondary siRNAs that will trigger very strong PTGS and systemic silencing. This has also been shown before, but what has not been shown before is whether the 22 triggers methylation, which is unlikely, but it is very likely that the double-strand RNA that is produced here upon transitivity initiation will methylate the coding region. Then the third class of small RNAs, the 24 nucleotide small RNAs, that when they are designed to target a coding region, an mRNA, they will be applied in the plant, loaded onto AGO4, but presumably they will not trigger PTGS, or they will trigger very, very weak PTGS, as we saw before. But what has not been shown is whether, as Michael said, whether the 24 nucleotide small RNA can target methylation here in the coding region. So we finished with the small RNAs, and now let's go to bigger, longer double-strand RNAs. We know from Michael Vassenager's lab last year that the delivery of long double-strand RNA compared to small RNAs is much less efficient. But we also know that eventually the small RNAs manage to somehow get in the cell and trigger PTGS, as has been shown in several cases. So what we do not know is how this process takes place. So we do not know the details of this mechanism. We do not know whether the double-strand RNA is processed in the nucleus where dicers are supposed to co-localize, or in the cytoplasm. And if they are processed by dicers, which type of small RNAs are produced? Unfortunately, no deep sequencing data have been performed in these studies that showed PTGS here. And, but since we have seen there are several cases where PTGS took place, we assume that 21 nucleotide small RNAs are produced, although they have not been detected. And since we have seen systemic silencing, we also assume that 22 nucleotide small RNAs also are produced. What has also been shown by Alexander Dubrovin and co-workers last year is that exogenous application of long double-strand RNAs in Arabidopsis triggered methylation of the coding region. But it was not clear whether the methylation of this coding region was induced by the primary double-strand RNA that was sprayed, or by the putatively 24 nucleotide small RNA produced, or by the secondary double-strand RNA that was formed upon transitivity. So we know that the long double-strand RNA can methylate the coding region, but we don't really know how this happens. So what has, we will continue with the long double-strand RNA, but now we will see what would happen whether a long double-strand RNA targets not the coding region, but the promoter sequence. To the best of my knowledge, this has not been tested before, but we will try to see what may happen. So similar to previous slides, the long double-strand RNA, we do not know whether it will be processed in the nucleus or in the cytoplasm by dicers, and also we do not know what type of small RNAs will be produced, if they will be produced. But even if they are produced, for example, if 21 nucleotide small RNA is produced here, there is no transcript available for PTGS, because here, this is the transcriptional start site, so presumably this part of the promoter is not transcribed. So if there is no RNA present, so the 21 nucleotide small RNAs will find nothing to cleave. But what has not been tested or analyzed yet is whether the primary sprayed long double-strand RNA or the 24 nucleotide small RNA that perhaps will be produced will trigger methylation of the promoter sequence. And now I will share some preliminary data that we have. So in order to analyze this situation, we use the 16C, the GFP expressing Nicotiana Methamiana plant, which carries this transgene, 35S promoter, a GFP coding region, and a NOS terminator. And we generated with in vitro transcription double-strand RNA targeting a big part of 35S promoter. So this is exactly here. So it targets a region of the promoter upstream of the transcriptional start site. And then what we did, we sprayed the plants and we made DNA extraction from the spray tissue, and after 3 days, we subjected this tissue to bisulfite sequencing to see the methylation of the cytosines. And what we could see in 3 days post-spraying is that we've actually found no methylation in this promoter sequence. So after 3 days, the double-strand RNA could not methylate the promoter sequence. So what you see here, these strange symbols are actually, they reflect cytosines the circles are cytosines in CG context, the squares are cytosines in CHG context, and the triangles are cytosines in CHH context. And if the symbols are open, then you have methylation. If the symbols are not filled, sorry, if the symbols are filled, you have methylation. If the symbols are open, you have no methylation. And as we see here, in both the control plants and the double-strand RNA plants, there was no methylation. This is insignificant, perhaps this is incomplete conversion. But when we took material 10 days post-spraying, this was the limit of before the tissue was completely screwed up and degenerated. We could see that the control plants showed no methylation at all, but in the sprayed plants, we saw very, very dense methylation, but not in all clones. I mean, if we focus here on plant 1, there are 10 clones, and we see that 4 out of 10 clones were completely methylated, and the 6 out of 10 showed no methylation at all. So this was an interesting part, suggesting that when some cells were completely methylated and some cells were not methylated at all, suggesting that perhaps the double-strand RNA was not present in these cells, or even if it was present, it could not be transported to the nucleus. And another interesting point was that the clones that we found methylation, this methylation was not spreading to the 3'. For example, if the double-strand RNA is mapping up to here to the 201 cytosine, so in this part of the promoter, there was no matching double-strand RNA, and indeed, there was no methylation. So this was quite unexpected, but it resembles a little bit the intron-based Rddm that Michael discussed before. But it seems that the double-strand RNA, or whatever was produced from this double-strand RNA, triggered very efficient Rddm in these regions. We could not analyze whether this methylation induced transcriptional gene silencing because the tissue was very, very degenerated. You could not really see fluorescence. And what we have not done, and this is very crucial, we have not performed deep sequencing, a small RNA sequencing from this data. So as I said, these are preliminary data, but they suggest that promoter Rddm may take place upon exogenous application of RNAs, but the efficiency has to be improved somehow. And let's go to my last slide. In order to improve this, the efficiency of promoter Rddm upon exogenous application of RNA molecules, we are now trying to optimize the method by on the one hand, to include carbon dots, like Michael said, or it was published last year. So that's why Michael, I asked you this question, whether you have tried it. And another point, which I think is quite critical, is that the long double-strand RNA needs, of course, to get into the cell, but this is only one part of the story. It also needs to get into the nucleus, because there is where the methylation takes place. And in order to achieve this, there are some RNA motifs which actually localize an RNA to the nucleus, like a CD boxing of SNO RNAs. And what we try to do now is to include these 25 nucleotide RNA NLS motifs to hairpin RNAs where they target endogenous promoter of legume plants. So actually with this manner, we would like to see whether we can indeed target endogenous promoters of legume plants for methylation. And in the end, our rationale is to see whether we can indeed silence some specific genes in legumes, and whether this silencing, this epigenetic silencing can be transgenerationally maintained. So I think I'm done.
Thank you, Athanasios.
Yes.
You have a question, Samantha?
So I see some, as it says, is there evidence for that the exogenous application, that the exogenously applied double-strand RNA is transported to the nucleus? No, there is no direct evidence for this. there's only indirect evidence based on the fact that we saw that the promoter was methylated. We assume that either the primary double-strand RNA was transported to the nucleus or some intermediate molecules produced by the double-strand RNA were transported. As I said, we have not performed deep sequencing, small RNA sequencing, to see what has happened, but we have really no clear evidence that the sprayed double-strand RNA was in the nucleus.
Rnas, may I comment also on that? Antje, there is probably indirect evidence because we think that the systemic silencing or the requires a nuclear step, but for all the virus people, there's a good indication that the if you use Wix for Rddm, which is possible, work done by Borkum, this indirectly shows that if you infect with the virus, RNA virus, the virus is not assumed to go into the nucleus. Nevertheless, if the virus contains a gene sequence of an endogenous gene, you will heavily methylate this by the virus, indicating that something from the virus produced by the virus is going into the nucleus to methylate the DNA.
Yeah, you're right. So we have some more questions. What was the difference between the different clones using Bethamiana sprayed? I think you mean the question means about the different clones in the bisulfite. So there are this. Ideally, each bisulfite clone represents a single cell. I mean, this is an assumption that we make. So each clone actually represents the different methylation, the methylation status of different cells. Then the other question is, are you mass producing small RNAs in bacterial or they were in vitro? They were in vitro synthesized. Why the 22s work better? It's, as we said before, it's because we think that the 22s induce better silencing because they initiate transitivity. So they produce secondary RNAs. Okay, so another nice question by Angie. She says that the 35S GFP of 16C is an artificial system. You're right. Do you think that this data, can be transferred to other gene systems? I don't know. I mean, you're absolutely right that the transgenic promoter is much more easily methylated than the endogenous promoter, but it can get methylated. So I think it will be quite challenging to methylate promoter, an endogenous promoter, and even more challenging to induce TGS of this promoter, because if methylating an endogenous promoter is quite hard, silencing an endogenous promoter will be very hard. So I don't know, but we're working on this, at least in legumes. Sorry. So it was the cleaning ladies who want to clean, if you're interested. Okay.
You have a few questions.
Yeah, there are several questions. And so transitivity in TGS, there was no transitivity, as we said. Gabi says, have you checked the methylation outside? No. Gabi, okay, so we show that we checked the methylation in the 3' region of double-strand RNA, but we did not check in the 5' region. I think we should have tried it actually, but I think that we will not find 5' transitivity in the 3', especially since we did not find the 3'. But it's a nice idea to check whether this happens. Okay, Alexandra said, yes, Alexandra says whether the tissue was injured and we could not see the GFP expression. Yeah, so yes, after 10 days, as you said, after 3 days, the tissue was still okay upon spraying, but after 10 days, the tissue started to degenerate. So I did not perform GFP mRNA analysis. I just tried to see the tissue under UV, but so I could not see GFP expression, but this does not indicate GFP silencing because the tissue was degenerated. Even in the buffer control, in the buffer sprayed plants, the tissue showed no GFP expression. So I think it's a physiological effect. It's not an RNA-based effect.
And may I also comment on that? You have to consider that this spraying method does not induce a very efficient silencing, and it's very different to the PTGS, to the RNAi mechanism, in the view that if you target one cell for post-transcriptional gene silencing, it will spread into the adjacent cells. If you make this for the epigenetics, there's a single cell where you introduce the methylation, and this single cell might be transcriptional inactivated, but then you need to have a microscope to follow this. And as long as we do not get a more efficient rate of introducing these TGS Rddm triggering molecules, it will be very difficult to see some effects on the transcriptional level, see the downregulation, the transcriptional silencing of the GFP. So you need at least, let's say, 30% efficiency of introducing this trigger RNA. This is different from all the PTGS. As you mentioned in your talk, Thanasis, we have to also be very careful if we target the coding region, because as soon as you put in whether a single molecule, as we did, it will induce transitivity, secondary double-stranded RNA production, and siRNA production. And we know that any activation of RNAi results also in Rddm of the coding region. So even if you take a short inverted repeat, the entire GFP sequence is methylated. And this is definitely not due to the effect of the primary introduced molecule, but to what happens after that. And this is, processing of the double-stranded RNAs 21, 24, and 22 nucleotide long siRNAs. And here we are, that's indicated clearly that it's always happening if you induce silent PTGS in the coding region.
The only difference would be that with the hairpin that is expressed upon agroinfiltration is that it's necessarily in the nucleus and then it goes back to the cytoplasm, and while upon spraying it's in the cytoplasm. So I agree that perhaps the methylation will happen because due to the secondary RNAs that are produced, but the difference between these two systems is that we do not know whether upon spraying what we spray is really present in the nucleus at one stage or another. So,
yeah, but if you spray at 22, it induces everything, yeah, systemic silencing, and which indicates that it's going into the nucleus. Otherwise, we do not see transitivity or systemic silencing, and also not the. And what we also know, if you apply double-stranded RNA into the cytoplasm, and the dices are in all also in the cytoplasm, it will produce 24s, and 24s are known to be loaded onto AGO in the cytoplasm and retarget to the nucleus. So also this will happen if you spray 24 into the cytoplasm, more likely, with the current knowledge we have.
Yeah, right.
I think it's good if you go on and then keep maybe some questions for the final discussion. I don't know.
So should we continue with the questions or not? I don't know if we have time. I wish we have. Okay, so another question by Alexander is whether the whole double-stranded RNA molecule is processed into smaller RNAs or whether RNAs are generated only to specific locations inside the double-stranded RNA. Okay, this is nice. I think, Alexander, you mean about the hotspot production of small RNAs. I mean, since we did not perform deep sequencing of the sprayed double-stranded RNA, we do not know whether first small RNAs were produced or which size they had, and whether they homogeneously map the double-stranded RNA. So I cannot really answer because we do not have data on this. So I think that perhaps the most of the double-stranded RNA would be processed to small RNAs, but it's just speculation. And actually, if I think we have 2 minutes, I may ask something about this. So the fact that some clones were completely methylated and the others not, I'm not really sure whether this reflects that the double-stranded RNA took some time to get in the nucleus, or whether the clones that methylated perhaps represent cells that were newly, upon cell division and the nuclear envelope lysis, they took up the double-stranded RNA. So what I mean is that I don't know whether the methylated clones reflect cells that were already established when I sprayed them, or whether these are the new cells after 10 days that were...
No, in the leaves, there's no cell division.
Yeah, but these leaves were very
small. You spray only adult cells, mature cells, and they do not divide, they enlarge, but they do not replicate. So yeah, it's unlikely to have a replicated nucleus there, an open envelope or something like that.
Yeah, but the leaves that are sprayed were not fully developed. They were really close.
Doesn't matter. They are tiny, but the total amount of cells is the same for tiny leaves and mature leaves. If they only expand, the vacuole gets larger, but you have no cell division. That's the problem why you cannot use a lot of these marker genes like PDS, because once it's established, you cannot reverse, and there's no cell division, so you will not see the phenotype. That's the problem.
So even a tiny leaf contains all the cells?
Yes.
Full.
Yes.
Okay. Okay. Good to know.
There was one other question: any transitivity in PTGS, and is TGS mobile? To give a clear answer, in our hands, it was not. We could not see transitivity, as also in the presentation from Thanasis, you could not see at least a downstream spreading. In our experiments, it also very sequence-specific TGS methylation. But whether if you, for example, apply 24s, whether they can move, as has been shown by Molnar et al., this we do not know. Maybe the 24 as nucleotide long as RNAs can move and may trigger something in other parts of the leaf or of the plant, in the roots or somewhere. So this we do not know.
Okay, so I think we can continue with the next talk by Alexandra Dubrovina, and from the Federal Scientific Center of East Asia Terrestrial Biodiversity in Vladivostok, Russia. So Alexandra, please.
Do you see my presentation? And do you hear me?
Yes, yes.
So I would like, I would be very glad to present you our work. And the full topic is physiological conditions and dsRNA application approaches for exogenously induced RNA interference in Arabidopsis. In our work, we are focused on the development of new eco-friendly approaches for changing various plant characteristics without genome modifications. So do you hear me in a good way? I mean the sound quality.
Yeah, I hear, at least I hear you very nicely, so I don't know the others.
Okay. And the focus of our work is to develop the development of alternative approach to transgenic plants, and we mainly focused on external foliar plant treatments with dsRNA. And the global aim of our work is the investigation of the possibility to regulate expression of plant endogenous genes via external application of dsRNA. It's known that plants can uptake extracellular nucleic acids, both RNA and DNA, for example, from viruses, microbial pathogens, giant plant cells. And in case this is double-stranded RNA, then the plant cell processes it and induces RNA interference or RNAi process to induce RNA-dependent gene silencing and lead to mRNA degradation or some epigenetic effects. And specific aims of this study. This study here, in this presentation, I would like to introduce you our recent publication. And this study aimed to analyze the importance of physiological conditions and different dsRNA application means for suppression of 35-driving niacin phosphotransferase 2 transgene in Arabidopsis. We decided to, in our first studies, we decided to focus on transgene because it's known that plant transgenes are more prone to silencing. So we believe this would be a good model system to train before any endogenous gene regulation attempts. And in this study, we analyzed the effect of plant age, time of day, and different abiotic stresses on the efficiency of foliar dsRNA treatments of Arabidopsis. We also tried different dsRNA application means. I will tell about them later. First of all, methodology of our study included generation of transgenic Arabidopsis. We used floral dip approach, agrobacterium-mediated plant transformation, and we obtained two independent transgenic lines, KA01 and KA02. The lines bear the NPT2 transgene under the control of the double 35S promoter. Then we quantified the NPT2 mRNAs in the transgenic lines of Arabidopsis by qRT-PCR, and we showed that NPT2 transgene is highly expressed in the two independent lines. And I would like to note that we worked with a single copy insertion lines, third generation and fourth generation. Then we focused on the synthesis and purification of in vitro transcribed NPT2 dsRNA. We used a commercial kit from Promega, and we synthesized. We created a large fragment of the NPT2 gene by PCR, and then followed the manufacturer instructions. Then we analyzed the dsRNA spectrophotometrically and measured it and analyzed it by gel electrophoresis to support its integrity and quality. The next step was to purify total RNA from Arabidopsis before any treatments. In each independent experiment, we used two plants. First plant was treated by water, and second by NPT dsRNA aqueous solution. For each RNA purification, we used we cut a single adult leaf, and after that we performed exogenous dsRNA treatments by 6 approaches. The water and dsRNA were applied on the whole leaf surface of individual Arabidopsis rosettes by 6 different approaches. And we used 100 microliters per individual plant, either water or 35 micrograms of dsRNA. In our previous study, we found that 35 micrograms per 4-week-old rosette is the most optimal concentration. Here, I would like, in order to show how we do this, just show you the video. First approach is spreading of the dsRNA solution by sterile brushes, just usual painting brushes, sterilized by autoclaving. We have spread the dsRNA from both upper and bottom side of each leaf, the whole rosette. The next approach was spraying. We used plastic containers just to simply spray the plant from upper and bottom sides of each leaf. And the next approach was infiltration. We infiltrated the solution onto the bottom side of each leaf, usual infiltration procedure. And we first approach inoculation. We inoculated the dsRNA solution using carbon containing inoculation mixture from both leaf sides, each adult leaf of a plant rosette. Needle injection, we injected the solution into each adult leaf upper side, usual syringe. And pipetting, we simply pipeted the solution onto each leaf of Arabidopsis from both upper and lower side. You can find the video in the supplementary material to our paper published in Plants. The next steps of our work included RNA isolations at 2 time points, 1 day post-treatment and 7 days post-treatment. So we finally isolated RNA before 1 day post-treatment and 7 days post-treatment. And then we analyzed the data using quantitative real-time PCR. Here I would like to introduce you the results on the effect of different dsRNA application means on the efficiency of NPT2 down regulation. So we used 6 application approaches: spreading by brushes, spraying, infiltration, inoculation, needle, and pipetting. And part A of this figure shows 1 line, and part B, 2nd line. And below you can see different time points of our treatments: 1 day post-treatment, 7 days post-treatment. White bars indicate control water treatment, and gray bars dsRNA treatments. And we found that spraying approach had the most pronounced effect on the NPT mRNA levels, the most pronounced down regulation effect, both 1 day post-treatment and 7 days post-treatment in 2 lines. However, spreading by brushes and pipetting also were efficient 1 day post-treatment. 7 days post-treatment, they means had a kind of limitation effect. Needle injection had a kind of middle efficiency, and infiltration and inoculation was the least efficient because of, I guess, traumatic consequences of the procedure. In all further experiments, we decided to use spreading of the RNA solution by brushes, since it was efficient in most cases and technically more comfortable for us compared to spraying, because we don't need sterile plastic containers. Here I would like to present the results on the effect of plant dsRNA treatment of different of plant age on the efficiency of dsRNA treatments. We use plants 2-week-old plants, 4-week-old plants, and 6-week-old plants. And we found that a considerable down-regulation effect was observed of only for 4-week-old plants 1 day post-treatment. While no considerable effect, as here, or even upregulation effect was observed for control water treatment. As for 2-week-old plants and 6-week-old plants, we did not observe any considerable down-regulation effect. We noticed that sometimes NPT expression was upregulated after simple water treatment. And we explained this either by impaired RNA degradation or elevated NPT2 transcription. In simple words, I would say that with time, plants accumulate NPT2 transcripts, mRNA transcripts. Here I present the data on the effect of time of day on the efficiency of dsRNA treatments. And I forgot to notice that in all experiments, in most of experiments, we used 4-week-old plants. For this experiment, we treated the 4-week-old plants at different times of the day, in the morning at 9 a.m., in the afternoon, in the evening, and at night. And surprisingly found that there is no inhibition effect, no considerable downregulation effect in the morning. However, in the afternoon, we see the downregulation effect, but only in one line, only in Ka01 line, and no such effect in Ka02 line. We found a considerable downregulation effect in the evening for both lines. However, only one day post-treatment. Also, we found a considerable downregulation effect both for one day post-treatment and 7 days post-treatment when we treated the plants at night. And I also would like to say that we purified RNA at the same time points, 1 day post-treatment and 7 days post-treatment. And we would explain this late daytime effect by possible importance of diurnal variation in plant transpiration rates and plant photosynthetic rates. Then we investigated the effect of different environmental stresses on the efficiency of NPT2 separation in Arabidopsis. And first of all, we applied cold stress. The plants were stressed at +5 or 2 hours after the dsRNA treatments. And here you can see Ka01 line and Ka02 line. And we found that essentially cold stress did not influence the efficiency of dsRNA treatments. It only improved it 1 day post-treatment in Ka02 line. Heat stress. The plants were stressed at 38 degrees Celsius for 2 hours after the dsRNA treatment. And we found that the heat treatment had a negative effect on the NPT2 expression. It appears that heat stress postponed the NPT2 downregulation. Salt stress. The plants were irrigated with sodium chloride solution for 2 hours after dsRNA treatment. And we found that sodium chloride did not exert a considerable effect on the efficiency of our dsRNA treatments. And soil desiccation. First group of plants were well watered just before the experiment, 1 day before the dsRNA treatments. And the second group of plants, water deficit, they were not watered for 3 weeks. So the soil was desiccated. And we found a kind of positive effect on the efficiency of dsRNA foliar application. However, it was more evident only for Ka02 line. It also appears for Ka01 line, but a more evident effect was for 0-2 line. Conclusions. Fuller application of the NPT2 dsRNA aqueous solutions reduced the NPT2 expression at a considerable level in the 4-week-old plants, while it was only limited in the 2- and 6-week-old plants. Thus, appropriate plant age is an important factor, ensuring a high efficiency of exogenously induced RNAi and Arabidopsis. A higher NPT2 suppression was detected at a late day period, being most efficient at night, which when we applied dsRNA at night, which indicated possible importance of diurnal variation in plant transpiration and lowers photosynthetic rates for active dsRNA perception and gene silencing. In addition, a more discernible downregulation effect was detected under low soil moisture conditions, which suggests that the dsRNA was absorbed more efficiently to intensify water supply. Treatment of adexal and abaxal leaf surfaces by brushes, spraying, and pipetting showed a higher NPT suppression, while infiltration and enucleation were less efficient. In conclusion, appropriate plant age, late time of day, low soil moisture, and optimal dsRNA application modes are important for exogenously induced gene silencing. I would like also to say that in our current studies, we are using this approach to downregulate several Arabidopsis endogenous genes, and this approach has an effect. This preliminary data, we are preparing it for publication now. Here, I would like you to introduce our research group. We are a group of scientists from Laboratory of Biotechnology, Federal Scientific Center of the East Asia Terrestrial Biodiversity. Konstantin Kisilev is the head of our lab, and I am the speaker. And this work was supported by a grant from the Russian Science Foundation. Thank you for your attention. I would be glad to answer your questions.
Okay, thank you, Alexandra. I think it's really important to see how the environmental conditions really influence this whole exogenous application plan. I think that you were really very committed in doing these experiments. I saw 3 o'clock in the night you went to make the analysis, so it's you were really devoted. So we have some questions by Gabi. If you have used any additives to the applied solution, and how do you explain the uptake of the double-strand RNA to the cell?
Could you please repeat the questions? I lost.
I don't know if you can also see it yourself in the chat, but she asks, did you use any additives in the applied solution of the RNA? And how do you explain the uptake of double-strand RNA to the cell?
We did not investigate the mechanism of dsRNA uptake, but some of our preliminary studies, we performed confocal imaging, and confocal microscope shows that dsRNA possibly is uptaken through the points for transpiration, and is transported by, sorry for my English, and is transported by phloem vessels.
But you have not used some carrier compounds, right, in the RNA, like carbon dots or dendrimers, or have you used something like this with the double-strand RNA, or it was a pure double-strand RNA?
Sorry, please, could you repeat?
I mean, if I understood Gabi's question right, she asked whether you applied with all these methods that you described, whether you applied double-strand RNA, in vitro transcribed double-strand RNA, or whether this double-strand RNA was conjugated to some carrier compounds, or like particles, or whether you use surfactants. I think you're right that you use carborundum in some stages, but did you use some other compounds?
No, we did not use other compounds. Only carbonodrum for mechanical inoculation. All other methods did not include any compounds. But we think that some surfactants can improve the efficiency.
Okay. And someone asked whether brush damaged the leaf cell. I mean, when you brushed the leaves, were they damaged?
No, when we use a brush, the leaves are not damaged. We use just regular painting brushes. They are soft and no considerable injury.
Okay. And if we don't have, I also have a question. If I got it right, you showed that the most potent silencing effect was 1 day instead of 7 days, right?
Yes, in most cases, yes.
Okay, but how do you explain this? I mean, is it a matter of the degradation of the double-strand RNA that was applied? Because since you targeted a transgene, I assume that this transgene would be target for RDR6 and transitivity initiation. So once you induce silencing in day 1 of this gene, then even if the initial input was degraded, then it would be a self-amplification loop would be initiated by RDR6. You know what I mean? Well, I mean, I would expect that the silencing state that you induced in day 1 would be continued, maintained forever, and not be diminished after some days. Would this suggest that there was no self-amplification mechanism in this NPT2 target?
Yes, we don't know exactly the reason for this, but this effect only in some cases is continued to 7 days, and we are planning to investigate the mechanism.
Okay. So we have some other questions by Ansi. Are you planning to extend your double-strand RNA uptake and gene silencing studies to other plant species besides Arabidopsis?
Yes, currently we are trying to perform similar experiments on tomato.
Okay. Barbara asks whether you observe downregulation of the target gene also when the plants treated with a control solution. I mean, did you see downregulation of NPT2 under control treatment?
It was in some cases, yes, but it was very slight, and we explained this by occasional effect of instability of a biological system. So it's just a kind of variability of biological system, which is not very precise.
Yeah, right. Okay. I think we have no further questions. So we can continue. I don't know if we have some more. Okay, no. So we can continue with the next talk by Sandra Correia from University of Coimbra in Portugal. And she will speak about the emerging roles of small RNAs in plant somatic embryogenesis. So Sandra.
Can you see my screen?
Yes, we see you and we hear you.
Okay. So good morning to you all, and first of all, I would like to thank the organizers for inviting us to present our work on a process that is in between a developmental process and a biotechnological one. So we are at the University of Coimbra in Portugal. You can see that the university, which is one of the oldest in Europe, it's a main feature of the city, and we are working around here at the Department of Life Sciences, where the which is here inside the botanical garden of the university. So it's a really nice place to work. And we are researchers at the Center for Functional Ecology, which works in a lot of different fields, but we are integrated in the research line concerning biodiversity conservation and ecosystem services, but mainly focusing on the use of plant biotechnology techniques such as in vitro culture. And that's why we have been working with this process. Somatic embryogenesis, as I told you, is due to the fact that somatic cells are able to be induced to regenerate an entire plant through a somatic embryo. This process is used as an experimental embryology system to address several questions related to embryogenesis. But of course, it has a very relevant biotechnological role, since it is a very useful tool for many important species, allowing for the clonal propagation of several important germplasm. And if you have liquid culture medium associated techniques, you can upscale that production through bioreactor techniques to upscale the amount of plants produced. And it is very important also because it allows you to preserve that germplasm and regenerate cryopreserved plants from those techniques. And it is a very important way to regenerate, transform the cells into plants for stable transformation. So it is a process with important applications for many species, from dicots to gymnosperms to monocots. And that's why it has been so studied. And recently, many reviews made by colleagues focused on the transcription factors and how those transcription factors are interconnected with auxin regulated genes, pathways, and also with epigenetic events. And small RNAs pop out in that question because they are connected to several stages of the process. Since the early stages of somatic embryogenesis induction through the maturation steps and then the germination of those materials. So this process has been studied in many crops, in many other species with from forestry also, and reviewed in recent papers, focusing mostly on the microRNAs targeting different transcription factors related to auxin pathway regulation. So it's interesting also to see that those it's coming up from the literature that there is a tight connection between several microRNAs and the auxin regulatory pathway, involving several stages, from the differentiation of the explant to until the stages where that embryogenic competence acquisition is acquired, and then how it is expressed when the competent cells develop into an embryo. And then that totipotency is expressed to a full differentiation process in which the embryos develop properly into a plant. And several different microRNAs have been correlated to these specific stages for different species. And based in this literature and working with our somatic embryogenesis system, we try to focus on those aspects of somatic embryogenesis regulation. We've been working with a Solanacea species, that is Solanobetaceum. This is a tree, but it has very well established in vitro culture protocols, such as somatic embryogenesis 1. So we can regulate, we can induce somatic embryogenesis from different explants, and in that induction conditions, we use high auxin and exogenous auxin and high sucrose concentration, and that explant differentiates into different cell lines, and those cells are callus, undifferentiated cells, and embryogenic masses that these ones can be transferred to a medium without auxin and develop into embryos and plants. So by controlling these conditions, these steps, we are able to reach a full plant, but still there are some kind of limiting steps that are common to this species and to many others. So the main questions still remain, despite the knowledge that we have today, the main questions still remain about the mechanisms that control how embryogenic competent cells are induced. And then when you have success in this stage, how is embryogenic competence maintained, since it's a problem that embryogenic competence is lost in subcultures? And then many times the true to type embryo development is difficult to achieve, and how can we improve the embryo quality? So these are still questions that deserve more attention, and we have been using our Tamarillo embryogenesis system to explore mainly the first two steps, and regarding the early detection of embryogenic competent cells, but also the comparing different cell lines to understand factors that maintain embryogenic competence. So we started by comparing these different cells. You can distinguish them by their cytological aspects. So the non-embryogenic cells, they are much larger with large vacuoles, and the embryogenic ones have a typical meristematic-like aspect. And by comparing different induced embryogenic and non-embryogenic cells induced with different exogenous auxins, we started by by profiling proteins differentially expressed, and we could see mainly metabolism-related factors, but also stress-induced proteins that were really differentially expressed between cell lines, and factors that are involved in protein translation, but also in chromatin remodeling and some stabilization. This was the first approach, but we could see it was evident from our analysis that embryogenic tissues were not homogeneous, and from a bulk analysis, some transcription factors might be a bit hidden. So we tried different approaches to get further into the research, and we use fluorescence activated cell sorting to reach different populations of cells. And we could further analyze those populations of cells. You can see here that from the same mass of embryogenic tissue, you can have really distinct populations of cells with different expressed transcripts. And we focused on those transcripts. So transcripts were related to auxin regulation, but also other plant growth regulators like abscisic acid and ethylene. We validated their expression, looking for the ones highly expressed in embryogenic samples, and through tissue culture and through the process, the entire process. Also, some transcription factors involved in chromatin remodeling were assessed using cell lines with different embryogenic ability. And we have been using this process to select putative important target genes to address. Also, we came across the hypothesis that by maintaining Kali in auxin for long periods of time, that Kali eventually lost the embryogenic ability. So we profiled the small RNAs in those samples, and we could see the different proportion of different classes of small RNAs in those samples. For instance, you can see that in long-term embryogenic Kali that is not embryogenic at this stage, you can see here this 24 nucleotide long up regulation compared to the other stages. And from this data, we are focused on several microRNAs with differential expression between tissues. And I must say that this is the work of Daniela Cordeiro, PhD. So she is evaluating the differential expression of the microRNAs, connecting them with different targets and validating their function in this loss of function during somatic embryogenesis. We have also done some previous work with some auxin-related genes and their regulation by specific microRNAs. It was also the work of Miguel Rito. He is now not working with somatic embryogenesis anymore because he is devoted to RNAi in plant pathogenesis, so an effect of iPlanta actions also, I guess. And but we did some studies regarding how specific target genes of microRNAs, such as microRNA 393, were regulated during the different stages of embryogenesis induction. So you can see shifts as you shift the culture conditions from auxin induced stage and when you remove the auxin and when you transfer the explants to light for embryo germination. And you could correlate strongly the effects of these microRNAs through the process and their effect on auxin-related transcription factors or receptors. So, to sum up what we have been studying with this system, so we based our research in the fact that you can induce cell lines with different competence abilities from the same explants. So with the same genetic background, you can have explants with different competence with different levels of totipotency, let's say. And you know the conditions that trigger that response, namely by the action of exogenous auxin, sucrose, and wounding are essential to induce it. So you can correlate, and we have data on the endogenous auxin levels and how they are correlated with the embryogenic callus formation. We also have data on proteins that are negatively related to the process, such as ribosomal RNA methyltransferases that are upregulated in non-embryogenic callus and that are a putative negative regulator of the process. And we could further identify other proteins related to the non-embryogenic samples, as well as we could detect proteins that are expressed in embryogenic cells and they are related to metabolism pathways and through the expression of heat shock proteins as well. Enzymes related to the ability of the dynamics of the plant cell wall were also identified, and then by a refined approach involving RNA-seq and fluorescence activated sorting, we could identify other transcription factors involved in the main processes that we are focused on. There are auxin-related transcription factors, that their connection with microRNAs, translation regulation, and of course, chromatin remodeling is a key step in these events. We have also data correlating the increasing of DNA methylation in explants submitted to long time exposure to auxins. So this is to sum up what we have, the potential of the system, and we are now at the stage in which validating this interaction could be these new tools of RNAi could be very effective in modulating this system. I mean, RNAi have been used to validate functions but not in a sense to modulate the process. And we are interested now in use those tools to modulate the process, since we know the wires now. So the purpose is to start cutting some wires to try to effectively modulate the process. So I would just like to finish by acknowledge the funding involved in this project. Several people from different labs, but mostly the students that work on these results, and George Canhoto, which is the PI of the laboratory. So, and thank you for your attention. I would be glad to answer some questions.
Thank you, Sandra. So I don't know, do we have some questions here? Yes, Gabi asks in the chat, how uniform are the calli, and are there critical few cells that give rise to the plant, or any cell can virtually turn into plant? I mean, how many, first, how uniform are the calli?
Yeah, regarding the first question, the calli is very heterogeneous. I mean, we have histological data on the, so a few cells on the surface of the explants respond and acquire that meristematic-like behavior. So there are not that many cells in, it's a restricted group of cells. And you can see a range of populations in tissues, embryogenic tissues. And the second one I could not.
The other question was whether, in principle, any cell can turn into plant, or are there some cells that have a particular characteristics that give?
Yeah, that's actually the main question, and the answer is no. Not every cell can eventually lead to a plant, And we have from many systems, we have already good data. That's what the main message I was trying to pass, that we are connecting the dots very well with model systems, and we identified several transcription factors, several microRNAs, how they are connected to auxin, how auxin is connected to epigenetic pathways as well. So we are connecting the dots, but we need efficient measures to modulate the system. I think I see from our work and from excellent work from colleagues that yeah, that step is still missing. So an effective modulation to target specific questions, but I think we're getting there now, and hopefully with these techniques of RNAi, like carbon dots, techniques to deliver RNA in a more efficient way than the traditional ones, could help to target specific steps of the process.
Okay. So do you have some more questions here? Okay, I have two questions. The first is whether you showed some small RNA data, that the peaks of 21, 22, 24 nucleotides. So these are bona fide microRNAs, or there are siRNAs and microRNAs?
Okay, we are still getting into that. So we are now focused more in the microRNAs. So because we were trying to more directly assess the ones connected to the transcription factors regulation from previous work. But we still need to look into that data. Yes.
Okay. And the other question is that you said that you will try to analyze the epigenome, the DNA methylation. What about the hydroxymethylation in these tissues? Because it's known that besides methylation, here we have also hydroxymethylation. How important do you think it is?
So far, the data that we have is in global methylation levels. We have not addressed hydroxymethylation yet, but and it's not that common to see that issue addressed, but I believe it should be important, yes. But we have no data on our system so far about that.
Okay, thank you. So, some more questions? No. Okay, then we'll thank you, Sandra, and we'll continue with Kriton Kaladidis from University of Crete in Heraklion. Hello, Kriton. And
hello.
He will speak about the role of RNAi machinery in viral and viral satellite infections. So, Kriton, please.
Thank you very much, Thanasia. Thanks very much, everybody, for being here. Can I share my screen? I think I can. Yeah, can you all see my screen?
Yes, we see it. Yeah.
Great.
Enlarge the full screen. Okay.
It's fine now, no? Okay. Thanks very much. So I'm going to talk about the understanding the role of the RNAi machinery in viral infections mainly, but also I will get a little bit into the role of the RNAi machinery in satellites that we're now starting. In this last project, we're a little bit behind our schedule because of the COVID effects that also affected us like everybody else, but I think we have some nice results in general. So this talk will be dedicated to Ricardo Flores. Whoever worked with the viroids ever would know him. He was a prominent figure in the field, but more importantly, he was a great person, a great teacher, and a great citizen. He was exceptional in all these aspects. He died a few months ago, so this talk is going to be dedicated to him. He's a pioneer in the viroid research. So viroids are small pathogens that infect many species. They infect only plants. We know nothing similar in other organisms. Hepatitis Delta reminds a little bit viroids, but has also some important differences. So Viroids are also are interesting for two reasons. One is because they are very interesting biologically, and I think I will convince you about that in a while. They do everything you will see without producing any protein, but they're also important because they infect species which are economically important, significant, and they cause significant economic losses. In some of these plants, like for example in tomatoes, it's a quarantine pathogen for some plants, and it's also devastating the effects it has on palm trees. So it is also an important pathogen for cultivations. What is amazing about them is that they do all this with a minimal genome. Their genome, an RNA genome, is 250 to 400 approximately nucleotides, and they do not encode for any protein. So anything they do it purely by their RNA sequence and structure, of course, secondary and tertiary. You'll see in a minute how this might look like. And they are grouped in two groups. One which is replicating solely in the nucleus, and one in the plastids. Today I'm going to only talk about the ones that replicate in the nucleus, with one exception. The fact that they replicate in there doesn't mean that they stay there, because you obviously understand that the pathogen needs to go from one side to the other, needs to travel, needs to infect cell to cell and systemically. So obviously it doesn't stay in the nucleus, it replicates there. And it replicates through pol 2, by the way, DNA-dependent RNA polymerase. Viroids are exceptional in various ways. One of these is that they are by far the smallest pathogen on Earth. They're smaller even than a prion gene. They're absolutely tiny, and everything they do, they behave like a real viral pathogen. That's why they're called viroids. Otherwise, they're very dissimilar to viruses. They do it with this minimal nucleic acid, which shows how potent structure is. Some people in the audience here will probably smile seeing this slide. This is by early work of Michael Vassenager, who gave a talk earlier here. Viroids were first for various reasons, including being the first autonomously replicating nucleic acid that was sequenced, but also because it was where Michael and the group of Sanger showed Rddm. It was there where it was first identified in viroid effects on methylation in plants in a Cell paper many years ago. They are quite astonishing. Here is the only slide I'm going to show you from a viroid replicating in the plastids. This is its structure in red here. It's RNA. It's this double-stranded RNA, a circular RNA. And this little pathogenicity region back here, this little blue area, is the one which is responsible for this huge effect you see here with the white leaves. Spontaneous mutations cause a loss of this pathogenicity determinant, and then it's latent. You have it in the tree without having any symptoms. And again, spontaneously, you revert back to this fantastic phenotype through this small alternation in its structure. So the structure is extremely potent and important. By the way, it's a small RNA produced from this region that causes this effect. The viroid we usually work with, but it's not the only one, is potato spindle tuber viroid. It's the first identified, and it's quite important. It's also a quarantine pest for many European countries. In the UK, you cannot even experimentally work with it, by the way. And it is a rod-shaped circular RNA that has lots of regions which are double-stranded, but also lots of regions which are not double-stranded. And many of these regions are important for all its functions, meaning its replication, its movement, its infectivity, blah, blah, blah. The questions we have had as a lab for a very long time now, as long as I remember practically, is looking at the interactions between viroids and the silencing pathways, and also identifying supportive host proteins. I won't have time to talk about both of them, only going to talk about the effect, its interaction with silencing pathways. We have some very interesting new results with the interaction with supportive host proteins, but this will be for my next talk. So I'm only going to talk about silencing pathways. I hope, and I think you know, how the silencing pathways work in plants especially, but in all organisms. This is a very nice review from Richard Carthew that summarizes what happens with any kind of double-stranded RNA in any kind of eukaryote. That's why it's so nice. So roughly speaking, double-stranded RNAs are a potent substrate for dicers to recognize them and produce small RNAs. These can be fully double-stranded or partly double-stranded, and the small RNAs produced, which again can be fully or partly double-stranded, form the sequence, give the sequence specificity to an Argonaut complex to go and do various things. Okay, so the obvious question is this structure here something that would be attacked by the silencing pathways? The answer is obviously is attacked. This has been shown already in 2001 from various groups, including the group of Martin Dabler, in which I was working at that time, in the sense that small RNAs from this variant were seen. But it wasn't obvious at all whether this is something that is devastating for the virus or if it's something that it just has to evade. Obviously, to some point, it did evade because it did infect plants. So there had been since many confusing, let's say, contradictory reports about whether the effect of RNAi is a positive or a negative one on viral biology. And as I think now, in retrospect, looking back, maybe Mikael can comment on that later, we can say that it's both. So viruses do take advantage, evolutionary have co-evolved with plants having RNAi, and they do fight back and they do take advantage, but plants also use it as a means of fighting against. That's almost what happens in any kind of cross-reaction between an immune mechanism and any pathogen that is still active and still able to infect. So to ask this question, we went to Nicotiana Bethaniana, and we did suppress each and every dicer and their combinations. So we went to the, this is a slide copied from a paper in the literature. It's not absolutely correct, but it's the most useful because it's very simplistic. So it helps me very much. It shows the actual dicers that we suppress and the pathways they take part in. So we went and by RNAi suppressed RNAi factors. Many people had complained to us in many ways about how is it possible to suppress RNAi with RNAi? Well, it is possible. If you suppress individual pathways, the other pathways take over. If you suppress multiple pathways, we now know that what is critical is that your Argonauts are loaded. And when your Argonauts are loaded, then you don't anymore need very much of dicer to do the job. So you can do RNAi because the Argonauts are loaded. So please keep that in mind. So we did we make our transgenic Bethaniana plants. We did our deep sequencing analysis. This was published a few years ago. We did suppress the dicers we wanted to suppress. We were not so successful in dicer 1, which is the one producing microRNAs. This is not surprising at all because, of course, you know that it's embryo lethal. So we did manage to get only leaky mutants. Whereas for the other dicers, the suppressions were very good. And of course, we did produce multiple transgenic lines for each suppressive event. We then went and looked to, this is work by Elena Dadami, by the way, a long time ago. She went to look at the specific, by Northens, at the specific small RNA species which represent, or let's say, are specific for each one of the dicers. You know that dicer 2 in plants produces the 22 nucleotide small RNAs, dicer 3 the 24 mers, and dicer 4 as well as dicer 1 produce the 21 mers. So in a very elegant series of Northens, by the way, Eleni and later Nadia showed that the specific suppression of dicers had a specific effect on the functional products of these dicers, which are the cognate small RNA species which correspond to these. Later, we did a combination of the suppression. We combined, we suppressed a combination of the dicers, and again, a combination of the small RNAs were missing in the respective dicer knockdown line. And this is what is shown here. So when you suppress multiple dicers, multiple small RNA classes are missing or are very strongly suppressed. You still see a few small RNAs because the dicers that produce them are suppressed, but they're not totally missing. And the Argonauts that do the suppression are there and loaded, but of course they have a turnover. So they need some small RNAs to be taken over and continue to do the suppression. And this was shown also again by transcriptome analysis, where we did see that the individual dicers were very nicely suppressed in the combined suppressed lines. Did these plants have a phenotype? Yes, they did. They had a much stronger phenotype than one is described in Arabidopsis, for Arabidopsis or for rice, by the way. I cannot give you many details because I don't have the time, but some of these mutants are quite unique. Like, for example, the combination of dicer 1 and dicer 2 gives totally unique phenotypes. This is a leaky mutant, of course, only a small partial suppression of dicer 1, but still gives very interesting phenotypes that I won't have time to talk about now. These plants were also interesting biotechnologically because by suppressing both dicer 2 and dicer 4, this is one of the lines of dicer 4-2 suppression, we were able to get very, very high levels of expression of agroinfiltrated proteins, in this case GFP, equal or even better than when you use P19. So you don't even need to infiltrate with P19 because you do get this very high suppression, and in some cases you don't want to co-infiltrate with P19. So these are very good plants to overexpress your RNA of interest as well. To make a long story short, because this was published quite some years ago, suppressing individual and multiple dicers at the same time, and then asking the question, what does that do to the viroid? The answer was quite surprising and took us quite some years to understand why. When anytime you suppress dicer 4, this is the main antiviral dicer, I remind you, this is the dicer responsible for RNAi. Anytime you suppress it, the viroid was the opposite of what we expected, was unhappy, was suppressed, was in low amounts. And we now think we understand why. This is because it is the combination of dicer 2 and 3 that fight back against viroids. And only when these two dicers are missing, then the importance of dicer 4 becomes obvious. When all three are missing, the viroids are really, really happy. So yes, dicer 4 does suppress the viroid, but it's hiding the role of dicer 2 and dicer 3, which are the main suppressors of viroid. So only when these two dicers are out, then you can see the effect that dicer 4 would have. And we have suggested in the past that viroids have evolved to bind primarily to dicer 4, and by this way evading dicer 2 and 3 suppression. By the way, viroids replicate in the nucleus. I remind you that this will be important information for the next few slides I have. So this is again the summary of all this. When you have a suppression of both dicer 2 and 3, the viroids are really, really happy. They really go up. And when you suppress even in addition to these two dicers 4, then the viroids are really, really in the best moment. Otherwise, dicer 4 covers the effect of dicer 2 and 3, and this is why evolutionists preferred to be digested against dicer 2 and 3. Now we have, with the help of Michael Vassenager and actually Thanasis Talakouras when he was there, we have generated mutants of dicers with CRISPR. We have already done this for dicer 2 and dicer 4. These have been now analyzed. We produce mutants which produce termination codons, but at the rather end, let's say, of the protein. So we were a little bit worried how successful these mutations were, although they do lose one of the two RNase 3 domains and the double-strand RNA binding domain. But I think we were successful, as you will see in the recent analysis for dicers 2 and 3. In both cases, the proteins are mutant. They are shorter than they should be. And in both cases, we do see the small RNA species, which are supposed to be suppressed, to go down in our CRISPR mutants. And now we have generated crosses also of these lines, and we're already analyzing dicer 4 mutants as well. And we're also generating multiple mutants at the same time. So CRISPR mutants are coming up, and at the moment they seem actually to be reproducing what our RNAi results showed. Now, this is unpublished stuff. When we went down to see what happens with the localization of dicers, these are Arabidopsis dicers expressed in the Damiana leaves. So we did see again that dicer1, as expected, was of course nuclear. This is not surprising. And the viroid infectivity did not change this localization. Again, you see it very nicely in the nucleus. So it stays where it is, and it doesn't seem to affect very much the levels of expression. Dicer2 is more interesting because it's in the nucleus, but it forms very often foci, where it concentrates in there. And again, PSDVD infection didn't seem to particularly change these. These, I think, some very nice pictures. Maybe you can see the foci in here. It's not uniform with the nucleus. So the regions inside the nucleus where dicer2 is concentrated. Now, dicer4 is more interesting. So dicer4 has two forms. It has a form which is cytoplasmic and a form which is nuclear. And what we found interesting is that the cytoplasmic dicer, when we infect with a viroid, it does get very often to the nucleus. In approximately half of the cells, when viroids infect, the dicer4 that is the dicer4, the cytoplasmic 4 of dicer4 seems to be transferred to the nucleus. Our speculation is that on the base of the model I showed you before, the viroid binds to dicer4 and somehow maybe functions as a nuclear localization signal for it and drives it into the nucleus. We don't know exactly how this happens. We don't know details about this. This is really fresh stuff from the lab. It's not published. This is what I showed you, and this is the whole story for dicer1, dicer2, dicer4, and dicer4 cytoplasmic, which in some cases stays in the cytoplasm, but in some cases, and only in the case of viral infection, goes to the nucleus. I left out what happens, what is the effect of dicer1 on viroids. We know from plastid replicating viroids that dicer1 is important in the phenotype. They do show sometimes because dicer1 produces a microRNA type, small RNA produced from viroids, and this has an effect on the phenotype. So we tried to ask the same thing in the Damiana. It was impossible because of the very strong effect it has on the phenotype on the plants. So we tried to skip the dicer1 suppression directly, which we only managed to suppress to a small extent. And instead we suppressed serrate, another member of the same pathway. And we asked what this suppression does to viroids. And what we saw, these plants, by the way, also have a phenotype. They have a very, very reduced, especially in tobacco and less in Damiana. They have a very, very slow development. This was also published a couple of years ago, 2 years ago now, I think 1.5 years ago, in Virology. By the way, the effect of serrate. And what we saw is that this is the levels of serrate. They were really strongly suppressed. We managed to suppress serrate much better than we did with dicer1. So we were more optimistic that we could see the effect this pathway has on the viroids. And again, to make a long story short, we did see an effect, or a strong effect, of serrate suppression on viroids. So indeed, it seems that this pathway is a pathway that the viroid takes advantage of. By the way, this is agro-infections, because when we did mechanical infections, when serrate is suppressed, and it's not suppressed to knockout effects, they are suppressed as a knockdown, by mechanical inoculation, we didn't manage to suppress these plants at all by viroids. So it seems to be necessary. Unfortunately, serrate doesn't do only microRNA production. It also has an effect on splicing. So it is possible that the effect we see is an indirect effect through some splicing effect. So this we cannot answer to. This is the effect I told you on mechanical infections, which we did not manage to get when serrate was there. Okay, so this is a summary of this part. So all silencing pathways seem to be involved in the responses to post-pre viroids. We didn't do this only with PSDVD, we did it with multiple viroids. We did with CVD and HSVD, by the way. It's more or less the same. In fact, with HSVD is even stronger, the effect. Dicer4 is relocated upon viroid infection, which I think this is an unpublished result, and I think it's quite cool.
Ich habe noch keine Zeit für die Maus, danke. Ich muss noch warten, bis das fertig ist. Ja, ich komme später. Alles gut.
So the microRNA pathway likely has a positive role on post-pre viroid infectivity, and we still need to identify what exactly the microRNA part of the pathway which is somehow affected. sorry, Thanasi. Am I over? Because there's a question.
I think I hear you. Yes, there is a question, but this question is for later. When
Okay, sorry. Okay.
But you may continue, yes.
Okay, because I still have 10 minutes now.
Yeah, sure.
Okay, so just a short introduction about the next question we had, which is what happens with satellites. There is very, very little known about how satellites are affected by the silencing pathways, and we have now. It is only known practically that the satellite RNA is also a target of DICEs. That's the only thing that has been published. These are the publications that are around for this. And we ask again the question, which of the pathways are the ones that fight back against the satellites? Satellites are actually quite an interesting story, I think. There are various types. Hepatitis Delta that I told you before is really more of a satellite RNA to the virus rather than a viroid. And it's quite interesting economically because some satellites increase the symptoms against the symptoms of the virus, the helper virus causes, like this satellite of CMV, but other satellites of the same virus ameliorate the symptoms. So they're also economically important because specific satellites have a positive or a negative effect on the actual effect on the plants. So we ask again the same question. We asked it with the TBSV and its satellites. We realized that TBSV, not surprisingly, suppresses our RNAi construct, so our plants are not so well suppressed. So what we did, we generated mutants of TBSV that lack a region, have a mutation in P19, and this mutation in P19 doesn't allow us our plant to be used in these experiments. And we have now started to analyze the effect that the different silencing pathways have on satellite infectivity. But this is an open question. I still don't have results to show. I can only say that DICE3 doesn't seem to affect at all viroid infectivity. And this is also work now done with DICE3 knockouts, the CRISPR knockouts. Okay, so CRISPR knockouts have now been incorporated in these experiments. Okay, so the production of CRISPR plants and the combination is underway. As I told you, DICE2 and DICE3 knockout plants are already out and they're functional. They do eliminate the small RNA species or reduce strongly the RNA, the cognate RNA, small RNA class that they are supposed to. And at the moment, I can only say that DICE3, even in the knockouts or in the knockdowns, does not seem to be affecting satellites. Satellite levels in the plant, at least in the case of the satellite of TBSV that we use. And as I told you before, DICE4 seems to be relocated by the virus. And with this, I would like to thank the people that have done this work. Mainly, it has been Konstantina Katsarou, a postdoc in the lab, which has worked on this all these projects for a very long time. Elena Dadami, that initiated in the lab a long time ago. And Nicoletta Kriou-Vassenager that has done the serrate experiments. And I would also like to thank you for your patience, and I take questions.
Okay, thank you, Kriton. So there is already one question by Cecilia. She asks, what is the difference between DICE4 nuclear and cytoplasmic forms? Is it just one small difference in the NLS?
There are splice forms. Yes, there are two splice forms with a difference in the one contains and the other doesn't, the NLS.
Okay, yeah. So are there some more questions here? Yes. So I have a question. It's really interesting, the fact that the PSTVD relocates DICE4 to the nucleus. You published some years ago that the VIRP1 protein is interacting with the viroid, and this is the one that actually transports the viroid to the nucleus. Could this be an indirect evidence that VIRP1 is interacting with DICE4?
Could be. We are working on that, actually. It's very difficult to tell. It's very difficult to work with the VIRP1 because it has a big part in the end, which is strongly disorganized. And it has an effect on phase separation. So it's a protein that is quite difficult to isolate and work with, but it could be that the three interact. In fact, VIRP1 is a protein that has a bromodomain, and we have localized it on the chromatin. So it has a BRD domain actually, and it goes there, and somehow we believe that it transports the viroid next to pole 2. How in this story DICER4 gets in, I have no idea.
So we have one more question by Gabi. She says that normally you do not find satellite RNAs together with TBSV. You may find DI particles of TBSV. So with what satellite did you work in combination with TBSV?
Oh, I don't remember exactly. This is a collaboration with Bari, with the CNR Institute there, that they do work with these satellites all the time. I don't remember which exactly satellite it is, but it's a satellite they have been working for years in Bari in the CNR, and that's where we got it from. This is not a, let's say, combination that we chose ourselves.
Tito, may I directly ask you, when we consider the dicer activity, it may act on the replication intermediates and it may act on the mature molecule. Do you think that BCL4 is also binding to the mature molecule but does not cleave it?
It's possible that it does, but it doesn't cleave it for sure, because we looked very extensively for an effect that it would have in the replication, so in the negative, so to speak, strand, during the replication in the concatameric form, and we could not see, with multiple ways, we could not see any effect. So if it does bind, it doesn't seem to be affecting it. So we have now placed our focus on other RNase 3 enzymes to be important in this role.
But if this is the case, then it may protect by binding to the mature molecule for the attack of dicer2, which may induce the transitivity or secondary.
That's exactly what we think.
Okay, then I agree with you.
Okay, so more questions?
You have a question from Gabi, I think.
I think we answered this question already.
Okay. The previous one probably addressed to Sandra. I don't know if you also answered this one to Maria. It's a name.
Yes, Maria, I think she asked Sandra, right? Yes. She asked that if I'm not mistaken, the monocots are more recalcitrant to somatic embryogenesis than dicots. Do you see any obvious difference on microRNA level or epigenetic level if you compare your system with monocot systems?
Okay, I should answer now. Sorry, Quentin, to
No, that's fine. That's fine.
Yeah, they are, and in fact, but the systems in which somatic embryogenesis is widely applied in monocots, like maize and rice. So there are many data on the microRNA regulation, inclusive many of the most validated effects come exactly from those crops, and so those comparative analyses between different distant phylogenetic related groups has been attempted, and in fact there are several families that are extremely conserved, from monocots to gymnosperms, but there is still very specific changes in those regulatory pathways between monocots and, for instance, dicots. And the system is, I think, is way better characterized in monocots and the interactions. So it depends a lot on the family of microRNAs you are considering. So I think in my perspective, the general answer is that it depends a lot, because there are specific variations, even though the mechanism is highly conserved.
Okay. So we have still some time. So if there are some questions for all lectures, so please don't be shy.
Well, it was really complex, I would say, a really interesting webinar demonstrating how complex is this system, but how effective can be if we can manage all the factors that are controlling this mechanism and the potentiality of this technology in controlling different mechanisms, not only and not only diseases. And I appreciate the fact that we combine all these different approaches. So for me, it was really an effective webinar. I don't know if there are some additional questions or comments related to this. Maybe it was so too difficult, too complex in some way.
Kriton answered, I think this refers to Gabi. Kriton says that we have worked with satellites L and B10 of TBSV.
So, well, my general question from all of you, what really general, so you may express, you already say many of your idea, but you may give a suggestion to all of us on what can be one of the critical steps that we should try to consider in improving our work for people that is working, trying to use the development of double-stranded RNA, possibly in the development of new product, double-stranded RNA based product. And what can be the main constraints? What can be the main limiting factors?
I think it's very, very, very important to demonstrate by model systems or reporter genes that what you want to do with a double-stranded RNA, if you apply this exogenously, that it's indeed doing what you think it should do. In other words, as long as you did not verify and validate that you induce for example, the RNAi machinery, you should be very careful. And the same applies to using double-stranded RNA for epigenetic changes. The Thanasis has preliminary data that indeed there is a very specific methylation that is homologous to the double-stranded RNA that he applied. This is a good indication. But we have to get more clear evidence by any sequencing technologies that we have to really confirm, yes, indeed, there is no other interpretation possible but the one that we think is working on that. And there people should be careful, in particular if you think about applications for the field.
Thank you, Michael.
If I may pick up Michael's point of view, which I totally agree, and I think that's why we are so focused in validating how these microRNAs interact with the targets and with the phenotypic responses that we are expecting in the regulation of the response in the embryogenic response of the explants. And I think only after we reach this point of validating their interaction, I think we get to the point that those technologies, the RNAi technologies that you are developing would be extremely helpful because because you are working in double-strand RNA delivery to the systems, for instance. And so if we come together now at this point, combining the knowledge we have from both approaches, yeah, I think the next step would be that to connect both lines would be very important, because in the case of somatic embryogenesis, it is an in vitro modeled process, and you can get really good answers from difficult explants for important species if you can get to that point. So it's my perspective, at least.
Thank you, Sandra. I don't know if there are other comments. From Antje, there is a question from Antje with respect to the application of exogenous RNA. How important is the possibility of gene methylation? Not very.
Okay, so I don't know if this. I think it's for me, perhaps. I don't know if Michael wants also to comment on this, but I'm not really sure what the question is. But if I got it right, I think that the gene methylation, whether it's in the gene body or in the coding region or in the promoter, is relevant because for two reasons. First, the gene body methylation may affect the transcription rate. So we do not know how the CG in gene body affects the transcription rates. And this is just having to do with the basic mechanism of how expression works. But actually, for me, it's very important the fact that perhaps the epigenetic modifications that we induce upon exogenous application may be transmitted to the next generation. I mean, this has some regulatory aspects that have to be taken into consideration. So I don't know if it...
In addition, I will comment on that. If we think about the double-stranded RNA applications for insect fungi, and then, of course, there is no effect in these pathogens. That's quite clear. It's only working if you target endogenous plant genes. Think about herbicides or something like that. And there, I think it's not very dramatic, as Tanasa said, if you target the coding region. But of course, if you would like to have it stable also in the next generation, and you think about regulatory elements, then it's of course relevant. Yeah, then you may produce. It depends what you would like to do. Would you like to have it stably inherited in plants, or would you like to have it in a period of the season only, or for a few weeks? Then you have to be careful. If you would have it transiently, then you need to consider epigenetic changes. But if it's doesn't matter, then of course there's no great impact.
And I would like to comment also something more on this, because I'm not sure if I understood correctly the question, but I would like to stress that even if you apply double-stranded RNA against a fungus or an insect in the plant, and this double-stranded RNA somehow manages to get into the plant nucleus and finds at least a 30 base pair homology with a plant DNA, as Michael Vassenager showed some years ago, then it may induce epigenetic changes. So it doesn't matter whether your double-stranded RNA is designed against an insect or a fungus or a virus. If this region has homology with a plant DNA, which is not very likely, but it's not unlikely, it may happen, then it will induce epigenetic changes. I mean, the minimum region, as Michael showed some years ago, was 30 base pair. So even a 30 base pair continuous homology between the double-stranded RNA, the fungal double-stranded RNA, and the plant gene will methylate the gene.
Okay, but this is a classical off-target effect.
Yes,
and
this is.
If we have the genome sequenced from the plants that will be sprayed, then one can try to avoid this.
Yeah, yes. So this is a suggestion to exclude the homologous region or the conserved regions, let's say.
Well, this is really important because then after it's going into the biosafety issue. Because if you have on target effect, even in terms of epigenetic response, it's something that
yes, but
consider also for that issue.
This can be excluded if the genetic information is known. This can be excluded, for example, by choosing another region of the plant of the fungal or insect gene. But this homework has to be done before the double-stranded RNA design.
But if I may, this is not very dissimilar with what happens with any kind of pesticides. You know, you never know what other effects it may have. You don't know. You cannot ever check every parameter before you apply. So, you know, people make some specific tests and then get a license and get out there and use it. And then 30 years later, and this happens all the time, somebody comes up and says, but it killed also moths, or it killed something else, or it had an effect, a side effect on the plant. So this is true for any new technology that you introduce in a living system.
Okay. So I don't know if you have some more comments.
No other comment, Athanasius, we can stop, we can end the webinar. I don't know if you have a final comment or.
Not really. I mean, I was covered with all the things that we discussed right now, and but I think it's for me, it was very nice to really touch upon this tripartite interaction between plant development and genetics and RNAi, and for me it was important to show this to share this message that that epigenetic modifications may take place in the sprayed plants, and this has to be more thoroughly investigated.
Absolutely.
And in addition, I would add that we are far away from understanding all the details. So I'm working for 30 years on Rddm, but nevertheless, there was not a lot of funding for these activities. And now in Germany, they started in the BMBF for such a program. And we were there discussing such a project, and decided that we need 80% of the money for basic research before we can do anything for applications. And you know, the BMBF in Germany is more interested in applications than in basic funding, but they agreed so that we are allowed to make 80% of basic research with the money that we may get for this. Far away. We have not, this is, I don't know why, we have not tested so far the application of 24 nucleotide long siRNA is what they do. Have not. But it's difficult if you use transgenes and you induce transitivity in systemic silencing, then you have an overlay with PTGS and you cannot get a clear answer. But this is in progress now. We are focusing on that, but little is known.
Well, it is one of the aim of this iPlanta to raise the knowledge on RNA technology and increase hopefully increase the potential interest from public institutions, maybe private institutions, to invest more for doing research. Clearly, I understand that many people like to have more results in terms of application, direct application, but as you said, I fully agree. We need to know much more things. So let's hope that also thanks to the cross-action and this network, people will be more aware of this potential tool, of the potential of this tool, and then will invest more for the future. I think we contributed to this, and that I'm, at least from my side, I'm really happy about that. And I want to thank Noel for the contribution given in all the work done for the webinar activities. Okay, if no other comments, and I want to thank really Athanasius, Michael, Sandra, Kriton, and also Alexandra for whole presentation today. Let's have the, as usual, the virtual group photo. I also have a coffee. If you want, we can have the coffee and virtual coffee. Let me prepare to take a picture. Please turn on your camera. Still some are missing. And few more. And Let me see if I get. Let me take another one. Okay. Perfect. Thank you very much. And goodbye to everybody. Let's hope to see again in the next, in another final conference. In some way, we are going to organize next. And have a nice day. Thank you.
You too. Thank you. And bye-bye.
Bye.
Bye-bye.
Thank you very much.
Bye.
Bye.
Bye.